All right, welcome to today's explainer, everyone. Let's just jump straight into it. Today, we're popping the hood on bioinformatics to look at something absolutely fundamental: the core differences between germline and somatic data analysis pipelines. If you've ever found yourself wondering exactly how raw genomic data gets translated into a life-saving clinical report, well, you are in the perfect place. So, let's dive right in.
Now, you might be wondering, DNA is just DNA, right? So why in the world do germline and somatic data need completely different pipelines? It's a great question. Well, when we actually look closely at the steps for processing and verifying data in these two very different clinical contexts, we realize they require totally different computational and physical approaches. We really can't just use a one-size-fits-all method here, mostly because the fundamental biology we're looking at, inherited traits versus acquired cancer mutations, well, it changes the math entirely.
So, here is our roadmap for today's explainer. First, we'll cover pipeline architecture differences, then dealing with data artifacts, and finally, we'll wrap up with wet lab validation strategies.
Okay, let's kick things off with Section 1, Pipeline Architecture Differences. Think of a germline pipeline like a really straightforward one-way highway. It's literally a straight line designed for a single input. You put the data in, and it travels straight down to the endpoint. But a somatic pipeline? That is a much more complex, paired path. It requires two simultaneous inputs and acts almost like this incredibly complex subtraction game. Let's break down that straight line for germline analysis. Because inherited mutations are present in every single cell, the process is super linear. Step 1, you just start with a patient's FASTQ file. Step 2, you map that to a reference genome to get your aligned BAM file. And step 3, you use a direct variant caller, something like GATK haplotype caller, to extract your VCF. It's direct, it's efficient, and it is absolutely perfect for inherited genetics. Now contrast that with somatic analysis. Somatic mutations are acquired, right? They're the specific drivers of a tumor. So to find them, the software has to analyze two files at the exact same time. Callers like MuTech2 require a paired run. So you input a BAM file from the tumor, sure, but you also have to input a BAM file from a matched normal tissue, like blood, from that exact same patient. Why do we do this? Well, because the software first has to identify all the inherited normal variants in that healthy tissue, and then literally subtract them from the tumor data. What's left over, the remainder, is your true somatic or cancer mutation.
All right, moving right along to Section 2, dealing with data artifacts. This part is actually fascinating. It's crazy how the physical handling of a sample literally alters the digital data. In cancer diagnostics, biopsy samples often come as FFPE blocks. That stands for formalin fixed paraffin embedded. The formalin is used to preserve the tissue, but get this, it actually damages the DNA in the process. This chemical damage introduces what we call FFPE artifacts into the data. Specifically, it creates artificial C to T mutations. And if you don't catch this, your pipeline is going to report a mutation that the patient literally does not have. Because of this, somatic pipelines require highly specialized algorithms designed to identify and filter out these exact artifacts. And that's a step that's just, well, it's generally not necessary in standard germline analysis. And it's not just chemical artifacts, it's also cross-contamination. In a germline pipeline, minor contamination from another sample might actually be tolerable, simply because the mutations you're looking for are just overwhelmingly present. But in somatic analysis, absolutely no way. It requires incredibly strict checks. Because tumors are so biologically complex, we're often hunting for mutations at a very, very low variant allele frequency, or VAF, sometimes as low as 5%. If your sample has even a tiny bit of contamination, it completely obscures those low-frequency cancer mutations. You could literally miss a critical diagnosis. This is exactly why tools like GATK's calculate contamination aren't just optional add-ons in somatic pipelines. They are the absolute mandatory first step before you can trust any of your downstream data. Which brings us to our third and final section, wet lab validation strategies. Here's the really crucial takeaway from all of this. The digital data directly dictates your physical lab strategy. For inherited germline mutations, your expected variant allele frequency is high, around 50 or 100%. So you can easily validate that with standard Sanger sequencing. But with somatic tumors, you're dealing with a messy mix of cancerous and healthy cells. The VAF drops way down to just 1 to 5%, and that completely changes the game in the wet lab.
Why? Well, get this. 15 to 20% is the standard detection limit for Sanger sequencing.
Let that sink in for a second. If your massive, complex bioinformatics pipeline accurately identifies a crucial somatic mutation at a 5% frequency, and you try to validate it in the lab using Sanger sequencing, the machine is going to completely miss it. The technology just isn't sensitive enough. It's essentially blind to the exact cancer mutations the somatic pipeline just worked so incredibly hard to uncover.
And this perfectly illustrates why somatic validation requires totally different technology. To confirm those low VAF results, the wet lab has to use absolute quantification techniques, things like digital PCR or droplet digital PCR. These are incredible tools that can detect variant frequencies as insanely low as 0.1%. They have the immense precision required to physically prove the existence of that tiny needle in the haystack that the somatic caller just found in the digital data.
So, to wrap this explainer up, I want to leave you with this quote. The biology of the tumor dictates both the digital code and the physical lab work required to treat it. What we've seen today isn't just a difference in software preferences, right? It's a testament to the immense precision required to actually understand human disease. Germline and somatic pipelines exist in two completely different worlds because the biological questions they're answering are fundamentally different. It really leaves us with a provocative question. As our pipelines get increasingly complex and our lab tools get insanely sensitive, are we truly ready for the level of precision required in modern bioinformatics? I hope this explainer helped clarify exactly why these pipelines differ and why getting it right matters so incredibly much. Thank you so much for learning with me today, and keep asking great questions.